As we continue on learning about cell energy, we're going to discuss now the processes that are involved in cellular respiration. As we said before, there are three processes involved in cellular respiration: glycolysis, the Krebs cycle or citric acid cycle, and oxidative phosphorylation, which involves an electron transport chain. In glycolysis, what we're going to do is we're going to take the glucose and we're going to break it down into two molecules of a substance called pyruvate or pyruvic acid. So we're going to, one molecule of glucose will be split in half through a series of steps using enzymes, and we're going to produce two molecules of pyruvate, two molecules of NADH, which are electron carriers, which will carry electrons to the final part of the process, and a net gain of two molecules of ATP. Kind of looks like this. Phosphate makes ATP and two NADH and produces two pyruvates. Now, the way we're going to make ATP is through a process called substrate level phosphorylation. Substrate, remember, is the molecule that an enzyme works on, and phosphorylation is the process of adding a phosphate to a molecule. So what we're going to have happen here is an enzyme is going to transfer a phosphate from some substrate to ADP and create or produce ATP. There are several compounds that will be formed in between the glucose reactant and the pyruvate product. These are called intermediates. We aren't going to spend any time talking about them, just know that there are some compounds in between. So here is a diagram showing the substrate level phosphorylation of ADP to ATP. We take a phosphate off this substrate. Both the substrate and ADP fit into the active site of the enzyme. We remove the phosphate and add it to the ADP. We produce ATP and release a product that now has only one phosphate instead of two. So we have two main parts, two main phases of glycolysis. We have an energy investment phase where we're going to put something into the process, some energy in, and the energy payoff phase, in which we're going to gain some ATP. So it's going to take two ATP molecules to energize the glucose molecule to go through the process of being split. And at the end, we're going to produce two NADH molecules, these electron carriers, and four ATP molecules. That means we're going to have a net gain of two ATP molecules. Since we're spending two here and gaining four here, then we have a net gain or a profit of two ATP molecules. So here's the process in short. Okay, in the first few steps, we're going to take the glucose and we're going to add, in a couple of steps, we're going to add a phosphate from an ATP here and another phosphate from another ATP here to end up with a product called glucose 1,6-phosphate. We have just used our two ATPs of energy. In the next part of the process, this glucose 1,6-phosphate is split into two 3-carbon intermediates, each of which has a phosphate on one end. Now we're going to show both phosphates as they travel through this process. In the next step, we're going to produce some NADH. We're reducing NAD plus to NADH, carrying some electrons and tagging along with the proton to produce our 3-carbon intermediate now that has two phosphates. The phosphate that comes here comes from a phosphate that is in the cytoplasm. It does not come from ATP. Next, we're going to produce the ATP and the phosphate. So in the pyruvate, we're going to remove one of the phosphates here off of one side and rearrange the molecule. Remove another phosphate off of the middle, producing our 1, 2, 3, 4 ATPs and our two pyruvate molecules. So our 4 ATPs here minus the 2 that we spent in the energy investment phase gives us a net gain of 2 ATPs, and we now have 2 pyruvates, 2 ATPs, and 2 NADHs from one molecule of glucose. Now, if oxygen is not present, this process will stop after a very short time because you have to have NAD plus for glycolysis to continue. And NADH is used in the step of the cellular respiration process that requires oxygen. If oxygen isn't present, some cells are able to do a process called fermentation. In fermentation, we're going to use the glycolysis process to produce ATP and some NADH, and then the fermentation process is going to have a way or give us a way to recycle the NADH back to NAD plus without oxygen so that we can continue performing glycolysis. Now, this occurs in organisms that do not require a lot of energy, things like bacteria and yeast. They're small organisms, not very energetic, and they can live without oxygen by doing this process. They don't produce a lot of energy, but it's okay if you're a bacterium or a yeast cell. If you're multicellular, like we are, and other animals and plants, you're going to need more energy than that from this process, and so you have to have the oxygen containing part of the process to produce the vast amount of energy that you can get from the glucose molecule. So our purpose here in fermentation is going to be to recycle NADH back to NAD plus so that we can continue that process of cellular respiration just with the glycolysis without oxygen. Now, your muscle cells and some kinds of bacteria can oxidize NADH through a process called lactic acid fermentation. In this process, NADH will be oxidized back to NAD+, and we're going to take pyruvate and change it into a compound called lactate or lactic acid. Now, this should sound a little bit familiar to you because lactic acid has the same root word as lactose, which is the sugar that is found in milk. Lactic acid is a milk acid, like lactose is a milk sugar. Milk isn't really involved with this, it's just the same compound that is found associated with many milk products. Now, the dairy industry uses this process, this lactic acid fermentation, to make food products like cheese and yogurt. It can also be used to make things like soy sauce, sauerkraut, kimchi, all of which are fermented products using lactic acid fermentation. So it has some usefulness in addition to recycling the NADH so that glycolysis continue. We can use the process to make food products that we enjoy eating. Here's what the process looks like. Our glucose, once again, is split into 2 pyruvate, producing 2 ATPs of energy and 2 NADHs in the glycolysis process up here. When oxygen isn't present, we need to recycle NAD+, and so this process takes those electrons and hydrogens back from the NADH and changes the pyruvate molecules into lactate, and it gives us NAD+ that can be used again in the glycolysis process to keep the process going and produce a little bit of ATP. Now, sometimes there's a different kind of fermentation called alcohol fermentation, and this is used in the baking and winemaking industries. When you bake bread, you use this. This is the kind of respiration that occurs when you make wine or beer and several other products like that. You use this same kind of fermentation. In this process, the yeast is going to oxidize the NADH back to NAD+, but rather than producing lactic acid, we're going to convert our pyruvate to carbon dioxide and a 2-carbon alcohol called ethanol. Ethanol is the alcohol that is found in beer and wine and other kinds of products like that. And the CO2 is going to produce bubbles or other things in the process. In baking, this is the process that happens. The ethanol cooks off as you bake the bread. The CO2, the carbon dioxide, is what makes the bread rise. Very minute bubbles of carbon dioxide get between the molecules in the bread dough and make it expand, and that's what makes bread rise from a dough that has been kneaded. Here's the process that happens here. Our glucose undergoes glycolysis, just like we saw before, producing ATP and NADH and our pyruvate molecules. In alcohol fermentation, 1 carbon and 2 oxygens are taken off of each pyruvate molecule and released as carbon dioxide gas. The electrons and protons from NADH are added back into the molecule to produce the ethanol, and then we end up with our NAD plus being recycled so that glycolysis can continue. Again, these are organisms that can survive without oxygen under certain kinds of conditions. Now, organisms that do not require oxygen are called anaerobes, and there are two main kinds of anaerobes. There are obligate anaerobes and facultative anaerobes. Obligate anaerobes have to be away from oxygen. They're poisoned by oxygen. They require anaerobic conditions. These are bacteria and other small organisms like that that live in stagnant ponds and deep soils, places that they're away from oxygen. And so they have to undergo a process like this in order to survive. Facultative anaerobes are able to live in either condition. If oxygen is present, they are able to do the entire cellular respiration process. If there is no oxygen, then they can undergo one of the two types of respiration, of the fermentation, to continue making some ATP from glycolysis. This includes yeast and a lot of different kinds of bacteria as facultative anaerobes. The facultative comes from the same root word that facilitated comes from, which also comes from the same root word as the Spanish word fácil, meaning easy, that a lot of you know. So these anaerobes have it easy because they can survive with oxygen or without it, whereas these, if oxygen is present, they cannot survive. Where we are what you call an obligate aerobe, that means we require the presence of oxygen. If we don't have it, we will cease to exist. Now, when you go to the next part of the cycle, the second part of the cellular respiration process is called the citric acid cycle. But before the pyruvate can enter the citric acid cycle, it has to be changed in a little bit of a way, okay? The pyruvate is going to be transported by active transport into the mitochondrion, where the other two parts of this process occur when oxygen is present. Remember that 2 molecules of pyruvate are produced for every molecule of glucose that enters the process. The pyruvate itself doesn't enter the citric acid cycle, but it has to be what's called a chemical grooming before it can do that, in which we're going to remove a carboxyl group, that is a carbon with an oxygen and a hydroxyl or OH group attached to it. We're going to remove that and release a molecule of carbon dioxide. The hydrogens and extra electrons are going to be transferred to NADH to make, NAD plus to make a molecule of NADH. And the 2 carbons that are left from the pyruvate are going to join with a molecule called coenzyme A. This is a cofactor that's going to form a molecule called acetyl-coenzyme A, which we abbreviate as acetyl-CoA. And acetyl-CoA is going to transport that 2 carbon molecule that's left over from the grooming process to the citric acid cycle. So the pyruvate has a carbon dioxide removed, produces another NADH, and coenzyme A will carry the other part of the molecule to the citric acid cycle. The citric acid cycle is also called the Krebs cycle, named after Hans Krebs, who figured out what was happening in this pathway back in the 1930s. It takes place in the matrix of the mitochondrion. Remember, the matrix of the mitochondrion is like the cytoplasm of the mitochondrion, and it occurs here because the enzymes that catalyze these reactions are found in the mitochondrial matrix. In this process, the citric acid cycle, we're going to complete the oxidation of the organic molecules, the original glucose, down to carbon dioxide, and it's going to produce a little more ATP and a pretty large number of NADH and FADH2 molecules. The NADH and the FADH2 are very important in producing most of the energy that results from the cellular respiration process. There's also a little bit more ATP produced by substrate level phosphorylation, and we'll talk about that as we go along. Here's the process. Acetyl-CoA delivers this 2 carbon molecule to the citric acid cycle. This is added to a 4 carbon compound that is already present there and undergoes a series of changes. In the process of those changes, each one of these carbons with 2 oxygens is peeled off of the molecule and released as a waste product. We're going to produce some NADH, 3 molecules of NADH, another molecule of ATP by substrate level phosphorylation, and a molecule of FADH2, another electron carrier. In the process of all this rearranging, we're going to reconstitute the 4-carbon compound that we started with over here before we added the 2 carbons from acetyl-CoA. So the acetyl-CoA is added to that 4-carbon compound, forming citrate or citric acid, which is where the cycle gets its name. Throughout the process of reactions, the citrate or citric acid is going to be broken down and degraded back to that 4-carbon compound we started with. We're going to release 2 carbon dioxide molecules and produce 1 ATP, 3 NADH, and 1 FADH2 molecule in the process. Now remember that we have 2 pyruvates that enter this whole process, 2 acetyl-CoAs. And so after 2 acetyl-CoAs go through the citric acid cycle, then the yield per glucose molecule from the citric acid cycle will be 2 ATPs, 6 NADHs, and 2 FADH2s. Plus, we're going to add the 2 more NADHs that are produced as we go from pyruvate to acetyl-CoA. So at the end of the citric acid cycle, in total, from the beginning of glycolysis, we've gained a total of 4 ATPs, 8 NADHs, and 2 FADH2s before we enter the last part of the process, which is the oxidative phosphorylation step. Most of the ATP production in cellular respiration occurs by means of oxidative phosphorylation. This is going to involve the transport of electrons through a series of carrier molecules and a process called chemiosmosis that produces the ATP and requires a supply of oxygen. This is the whole part of the process that requires oxygen and is going to recycle all of the NADH and the FADH2 back to their ground state so that they can pick up more electrons and continue the cycle. The electrons from the NADH and FADH2 are going to travel down the electron transport chain. The electron transport chain is a series of proteins embedded in the inner membrane of the mitochondrion. At the end of the chain, the oxygen is going to pick up the electrons and some hydrogen ions or proteins to form water molecules. The energy that is released by the reaction of moving the electrons from one protein to the next is going to be used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space, the space in between the 2 membranes of the mitochondrion. In the process of chemiosmosis, the hydrogen ions that have gathered there in the intermembrane space have developed potential energy like water behind a dam or water in a water tower. And as they diffuse back across the intermembrane from an area of high concentration to an area of lower concentration, they're going to pass through a molecule complex called ATP synthase. And as they do that, it's kind of like water passing over a turbine in a dam. It's going to generate that as the turbine generates electricity. In the process of chemiosmosis, when the hydrogen ions come back through the ATP synthase, it's going to generate ATP. So we're producing energy by the passage of these protons through this molecule that acts kind of like a mill to produce the ATP. So here's what we have. We have all these protein complexes. These are in the inner membrane of the mitochondrion. The NADH and FADH2 deliver those high energy electrons to these protein complexes. Each one of these 3 protein complexes that are shown here with a Roman numeral 1, 3, and 4 are proton pumps. This is an active transport process, and the high energy electrons here give enough energy to pump a couple of hydrogen ions into this intermembrane space. The hydrogen ions are going to collect here in this space, again, kind of like we're pumping water up into the water tower to have potential energy until we open the faucet to allow it to come back in. So the electrons are passed first from one molecule to a second to a third, and eventually we've reduced to their ground state or the lower energy level. At this point, they can be transferred to the highly electronegative oxygen molecule that is present here. Some of the hydrogen ions that have diffused back through the membrane can also add to this to produce a molecule of glucose. This part, the electron transport chain, is the flow of electrons that end up as as part of the water molecule being picked up by the oxygen. Meanwhile, we have all of these hydrogen ions that have collected in the intramembrane space. They're going to diffuse back through ATP synthase in the process of chemiosmosis to produce ATP. And that's the process of oxidative phosphorylation. So now, at the end of cellular respiration, from the glucose molecule undergoing glycolysis all the way through oxidative phosphorylation, we have produced a total of 2 ATPs in glycolysis, 2 ATPs in the citric acid cycle, and about 28 ATPs through chemiosmosis to produce a total of about 32 ATP molecules. We've also produced carbon dioxide, 6 molecules of carbon dioxide, 6 molecules of oxygen, and that gives us the products, the final products of the cellular respiration process.